Hello, my name is Cade Speed. I am a senior at Texas Tech. I am a student worker at Dr. Zhao's lab at the Institute of Genomics for crop abiotic stress tolerance. My presentation today is about genotyping sorghum mutants for gene function studies. Sorghum is the fifth most cultivated cereal crop worldwide and is considered to be an increasingly important future crop for both food and biofuel supplies. It is a heat and drought tolerant crop. Lubbock and the surrounding area's hot and arid climate provides an excellent growing habitat for sorghum, and it is an economically important crop to the Plains region. So why study sorghum genetics? It is critical to understand the genetic regulation of important agronomic traits for the application of modern biotechnologies to breeding. The Zhao laboratory seeks to study the effects of abiotic stress on sorghum. Abiotic stress is the negative impact of non-living factors on a living organism. For example, temperature, water availability, and UV radiation. As the climate changes and water resources become more limited, a more heat and drought tolerant sorghum variety is highly desired. How to begin studying sorghum genes. The Zhao lab is using a large sorghum mutant population to dissect traits related to abiotic stresses. Mutant populations have been shown as great material to study gene functions in plants. The mutant sorghum population was provided to the Zhao lab by the USDA Agricultural Research Service. Genotyping is the first step of studying the mutant populations as well as important technologies in molecular breeding. This genotyping project will design and test molecular markers, CASP, for important genes related to abiotic stresses. Competitive allele-specific PCR, or CASP, is a homogeneous fluorescence-based genotyping variant of polymerase chain reaction. It is based on allele-specific oligoextension and fluorescence resonance energy transfer for signal generation. The genotype-confirmed mutants will set up the foundation to study the regulation of these traits. The markers will also have the potential to be applied in breeding. The Zhao lab studies functional and comparative genomics. Functional genomics describes gene and protein functions and interactions. Functional genomics uses a genome-wide approach and focuses on gene transcription, translation, regulation of gene expression, and protein-protein interactions. Comparative genomics compares the genomes of different species in order to understand the differences between life forms at a molecular level. Sorghum genomes are being compared with rice and maize genomes to help better understand the unique characteristics of these crops. A gene of interest is the sorghum homologue of the rice gene SLG1 gene. SLG1 plays a key role in the response of rice plants to high temperature stress at both seedling and reproductive stages. The genotype-confirmed sorghum mutants will be used to test if this gene has the same function and heat tolerance. So some hopeful research outcomes will be improved heat and drought-tolerant sorghum varieties through breeding, lowered environmental impact with more agriculturally sustainable sorghum, more economically viable sorghum in this region for farmers, and increased knowledge in the field of crop genomics and abiotic stress to further combat climate change for the future. Thank you guys.